myself M. Nitya, working as Assistant Professor in the Department of Biotechnology, K.S.Rangasamy. So today we are going to see a lecture topic on somatic embryogenesis. So before getting into this topic, we must have to know what is somatic embryos. Regarding the embryos, there are two different types. One is said to be as a zygotic embryos and the other one is said to be as a non-zygotic embryos. And regarding the first category, that is the zygotic embryos, they are formed by the zygote or by the reproductive cells. Okay. So regarding the second category, that is the non-zygotic embryos or the somatic embryos, they have been obtained from the somatic cells. In short, we can define this somatic embryogenesis as the process of obtaining the embryos from the somatic cells. Okay. So before getting into this topic in depth, we must have to know what are all the different types of somatic embryogenesis. So regarding the somatic embryogenesis, they have been classified into two different categories. One is said to be as the direct somatic embryogenesis and the other category is said to be as the indirect somatic embryogenesis. Okay. So first let us know what is direct somatic embryogenesis. So the name itself implies that here we are going to obtain the somatic embryos straight away from the explant. Okay, so we are here in case of this first category, we are going to obtain the somatic embryos straight away from the explant. That is said to be as direct somatic embryogenesis. And how this is possible by means of this method is that already the explant contains a specific cell which is said to be as the PEDC cells. Okay, so let me abbreviate what is PEDC cells. This can be said to be as the pre-embryonic determined cells, or else we can say this is the pro-embryogenic determined cells. Okay, so because of the capability of the presence of this PEDC cells in the explant have the ability to develop the somatic embryos straight away from the explant. Okay, so this category is said to be as the direct somatic embryogenesis and the second category is said to be as the indirect somatic embryogenesis. So the name itself implies that here the somatic embryos have been developed indirectly from the callus. Okay, so first what we have to do is we have to isolate the explant, and after isolating the explant we have to go for surface sterilization, and once the process of surface sterilization have been completed, we have to inoculate in a suitable media, especially or specifically we have to use the MS media, and after that we have to induce the callus. Okay, so from this callus what happens is that the embryos will start to initiate. Okay, and if you are culturing the somatic embryos in a suitable media, your whole plantlet have the ability to regenerate. Okay, so this is the meaning for indirect somatic embryogenesis. So let me define what is this indirect somatic embryogenesis once again. So if you are obtaining the somatic embryos indirectly from the explant via the callus, it is said to be as indirect somatic embryogenesis, and this is possible because of the presence of a type of cell which is said to be as IEDC, which means that induced embryogenic determined cells. We are going to induce the explant to produce the somatic embryos by applying suitable hormones, growth hormones. Okay, so the next we can see about for indirect somatic embryogenesis, two distinctly types of media can be used. One media for initiation of the embryos and the second for development of the cells in the embryos. Okay, so in case of the indirect somatic embryogenesis, we have to prepare two different types of media. One media for initiation of the embryonic cells and the other media for the development of the cells into embryos. Now let us see what are all the stages of somatic embryogenesis. Once the process of somatic embryogenesis have been developed in the media, it requires three stages, or it will be passing three stages. They are, the first one is callus, the next one embryo development and maturation, and the third step is said to be as the plantlet formation. Okay, so once this process is going on, they will be going for the process, or they will be attaining the stages of 4, and they are said to be as globular stage, heart stage, torpedo stage, as well as the cotyledonary stage. Okay, so once the process of somatic embryogenesis have been initiated from the callus, there are 4 stages. They are said to be as globular stage. This is the first stage, and once the globular stage have been attained, they will be passing to the next stage, which is said to be as the heart stage, and the next one is said to be as the torpedo stage, and finally they will be attaining the cotyledonary stage, and finally we can able to obtain the plantlet, and once the the plantlet have been developed, we can go for hardening it. So generally, before seeing about this procedure from Dacus carota, we must have to know how we can perform the process of surface sterilization. So first what we have to do is we have to excise the explant from the natural environment. Okay. So once we have obtained this explant, we have to go for washing in our laboratory conditions with the tap water. So once the process have been completed, we have to go for washing with the distilled water. So once this process have been completed, we have to wash our explant with the detergent. So most frequently we are going to use the detergent like Tween 20 or Tween 80. So once this process have been completed, we have to go for the next stage, that is we have to go for surface sterilization with the help of babystin. Okay, so naturally this babystin will be acting as a fungicide. We have to add few drops of babystin inside our explant, and we have to add distilled water also, and we have to shake vigorously until our tissues have been completely immersed on this babystin. Okay, so once this process have been completed, the next process is that we have to go for adding, or we have to treat this explant with 70% ethanol. So this 70% ethanol will be having the germicidal activity. So once this process have been completed, then we have to treat our explant with 0.1% mercury chloride. So this we have to perform inside the laminar airflow. So we have to expose the explant in this mercury chloride for a period of 60 seconds, and then finally we have to trim our explant, and then now our explant is ready to inoculate. Okay, so let me sum up once again regarding the general protocol of surface sterilization. So first thing what we have to do is we have to go for excising the explant from the natural environment. So once we have obtained the explant, we have to rinse the tissue with tap water for several times, and then finally we have to wash our explant with distilled water. And once this process have been completed, we have to go for adding a little drops of Tween 20, that is a detergent. So this detergent have the ability to kill the germs, or and it also have the ability to remove all the dust particles that are present on the explant. Okay, so the next is we have to go for washing our explant with babystin, and after treatment with babystin, we have to go for treatment of our explant with 70% ethanol, which will be having the germicidal activity, and the application of this babystin is for eradicating the fungus that are present in the explant. Okay, so once the process of 70% ethanol treatment have been completed, we have to treat our explant with 0.1% mercury chloride. So this is the final step. So we have to expose our explant for a period of 60 seconds, and after that we have to remove our explant, and we have to drain all the water particles that are present on the explant, and we have to trim it. Okay, so once the trimming process have been completed, we have to inoculate this explant in a media that have been supplemented with suitable growth hormone. So now let us see the detailed protocol of how we are going to get the somatic embryos by using the plant Dacus carota. So the leaf petiole generally in the size of 0.1 to 1.5 centimeter, or we can go for taking the root segments from 70-volt seedlings from storage root, which can be used as an explant. The next step, the explant have been surface sterilized by following the protocol what I have said previously, and they have been washed with ethanol for 30 seconds, or else we can go for a period of 60 seconds, and then by sodium hypochlorite. So the sodium hypochlorite will be acting as a detergent, and finally wash several times with distilled water, and then this explants are individually placed on a semi-solid MS media, which will be containing a growth hormone of 2,4-D, which will be acting as a auxin. So the auxin have been added in the concentration of 0.1 milligram per liter, and we have to supply 2% of sucrose to this medium. These cultures are incubated in the dark. So in this medium, the explants produce sufficient callus tissue. Okay, so this is the medium what we can go for initiating the callus. And the next step is after 4 weeks of callus growth, again we have to transfer this callus tissue to a sterilized flask containing 20 to 25 ml of liquid media with the same composition that we have previously used for initiating the callus. The presence or absence of light is not critical at this stage, but previously we have to incubate the culture in a dark so that callus will be initiated. Okay, but for this step we should not provide any light, or else even if we provide any light also the process will be continued. Okay, the next one, cell suspensions are subcultured every 4 weeks by transferring 5 ml to 65 ml in fresh liquid MS media. Then to induce more uniform embryo population, we have to go for in need of a sieve. So the size of the sieve is 74 micron. So this sieving process have been used for selecting the proper size of the callus, and portion of sieve cell suspensions are transferred to 2,4-D, that is the oxen. Free liquid medium or cell suspensions can be treated in semi-solid MS medium which will be devoid or lacking of 2,4-D. The next step, after 3 to 4 weeks, the culture would contain many embryos in different stages of development. So we know the different stages of development as we have already seen, that is globular stage, arch stage and the terminal stage, and finally we can able to see the cotyledonary stage. Okay, so they will be passing these 4 stages of growth, and finally the plantlet or transferred to the vermiculite for subsequent development. Okay, so this is the general protocol of how we can go for inducing the somatic embryos by using the explant Daucus carota. So this table provides a differentiation between the organogenesis as well as the somatic embryogenesis. Regarding the definition for organogenesis, this is the development of organs from an embryonic cells, and regarding the somatic embryogenesis, this is the process of development of somatic embryos from the somatic cells, and let us see how it occurs. Regarding this organogenesis, there will be less natural, whereas in case of somatic embryogenesis it is an artificial one. Either this organogenesis may be artificial or else it may be of natural, whereas in case of the somatic embryogenesis they will be occurring in nature, artificial. So where can the process be seen? So the process of organogenesis have been seen in both animals as well as the plants, and in case of somatic embryogenesis they have been seen only in case of the plants. And regarding the outcome of the process of organogenesis, an entire plantlet with shoot and root is formed in case of organogenesis, and only the somatic embryo is formed in case of somatic embryogenesis. And its association with maternal tissue, roots and shoots have a strong association with case of organogenesis, whereas the somatic embryos are not in vascular tissue in case of this somatic embryogenesis. So this is the diagram which illustrates the procedure for inducing somatic embryogenesis by using the Daucus carota. So first we have to excise the explant, suitable explant, and after excising the suitable explant, we have to inoculate this in a media that have been supplemented with suitable growth hormone in MS media. So after that we have to incubate this in dark. So once we are incubating this in dark for a period of 4 weeks, we can able to see the development of callus. So once the callus have been developed, again we have to subculture this in a suitable media which will be devoid of 2,4-D, so that again we have to go for an incubation period of 4 weeks, so that what happens is that we can able to see many embryoids in the media. So once the embryoids have been formed, we have to isolate that embryoid and we have to transfer this in a suitable media that have been supplemented with suitable growth hormone, and final stages we have to go for acclimatization or hardening. Okay, so in this way we can able to develop a full plantlet with the help of somatic embryos. So now let us see some of the important advantages of somatic embryogenesis. A huge number of embryos can be obtained by the process of somatic embryos. The development and development stage of somatic embryos can be regulated, and this process of embryogenesis can be monitored easily. And regarding the significance of somatic embryogenesis, we can go for production of artificial seeds or the synthetic seeds by using the somatic embryos. and higher rate of propagation have been obtained by using the somatic embryos, and they will be apt in suspension culture, that is a liquid media. And the most important significance of why we are going for the somatic embryogenesis is that it is a labor saving process. So what are all the fields we can go for applying this process of somatic embryogenesis? For in vitro selection we can go for this somatic embryogenesis, as well as for in vitro conservation we can go for using this process of somatic embryogenesis. And if we want to go for getting many number of explant, we can go for this somatic embryogenesis. That is, large scale propagation of the explant can be achieved by the process of somatic embryogenesis, and the genetic transformation have been successfully achieved by the process of somatic embryogenesis. And regarding the disadvantages of somatic embryogenesis, low number of field plantlets and highly genotypic dependent plants have been produced. So this have been considered to be one of the main disadvantages of why we are not going for somatic embryogenesis, and inability to produce somatic embryos from mature seeds. Okay, so if you are in many of the plant species, we can't go for isolating the matured somatic embryos. So this have been considered to be one of the major disadvantages of why we are not going for the process of this somatic embryogenesis. And gradual fluctuation and eventual decline in embryogenic culture productivity, and thus somatic embryogenic cultures from seeds or seedlings have unproven genetic value, and long life cycle may show genetic variability or new mutations at the later stage of development. So these are all the major disadvantages of why we are not going for somatic embryogenesis. Students, I think this lecture will be very useful for you. Thank you.